Good morning or good afternoon, everyone, whoever is looking to this talk, short talk, which is associated with the main course, which is about analyzing data in the cloud or in distributed environments, compute environments using Nextflow with Quantms. This talk is only about the configuration file, the main file we use to launch our pipeline to describe our data. My name is Perez-Riverol. I am the team coordinator at European Bioinformatics Institute. Um, what is actually SDRF? SDRF is file format that we developed last year. It's published in Nature Communication about how to describe the sample metadata and its relation with the data files. This actually um, came from a really old problem in proteomics, which is basically in proteomics in the archivals. Most of the data that is deposited are the instrument file, the raw files, the result files, and intermediate files, including also plots and configuration files. And they were also is also deposited the data set in general information, like a description of the title, abstract, protocols, speeches, and so on and so forth. But for a long time has been missing the relation between these instrument files and which sample was captured on each file. And that's what we want to model it here. Let's call it, this is the file format that try to model it the experimental design, but it's also capturing a lot of metadata around the samples and a lot of metadata around the file itself. I will, in this short presentation, I will try to explain what are the main advantages of this file format, and I actually will put some examples of my own annotated data sets and how this data set how are triggered with Quantms, which is the main objectives of this course. What are the use cases we want to support? We want to support with the file format, with SDRF, which is a tab delimited file format. We want to support to describe the relation between the sample and the files. The file format is actually enable the user, bioinformaticians, mass spectrometrists, biologists, to describe each sample in which file it is contained. File right now is mainly about the output of the instrument, the mass spectrometer. We want to make this file format actually easy for annotation, that someone with no expert in bioinformatics can open their own Excel software in their own computer, a TXB, and actually start annotated. It should be compatible with other file formats, and really, really important, it should be ontology based. What is an ontology? Okay, every term, we want that every term in the SDRF is actually a controller vocabulary term. Like if we describe, for example, what type of instrument, we want that all instruments of the same model are called in the same way. We don't want, for example, someone call it LTQ orbitrap and somebody else call it LTQ/orbitrap. No, we want the same term for the same concept. And that's what ontology based mean. We actually use ontologies to describe not only the properties, but also the values of the properties. Why SDRF? Well, we actually look into multiple options, and SDRF is actually not new. SDRF is actually came from the Mainstab for the microarray and RNA-seq data community. It is part of the Mainstab file format, which is extensively used in transcriptomic and RNA-seq. And the main idea is that we want to reuse the file format for in the future when someone actually performs a multi-omics experiment and they have their own RNA-seq data deposited in Array Express, and then they have their own proteomics data deposited in Pri. They actually use the same file format to describe the file. And there is extensive tooling for validating Mainstab and SDRF, and we will talk a little bit about that in the last slide. how an SDRF look like, okay? An SDRF is, as I said before, has three major parts, I would say, or components. It's a tab delimited file format, where it's basically meaning a table where every column corresponds to a property of the sample of the data file or the factor value, which is actually the condition, the variable under study. Let's say when you try to study in your experiment, like phenotype, like you, for example, you are, in this case, you have a cancer study, and then you have some control variable and then some primary tumor. This is called, for example, we call it here the phenotype. This is the variable you want to study. And each value of that variable is basically the value of that condition for this particular sample in this particular raw file. It is also important to know that this is the relation. In this case, for example, in the first part of the table, you have sample 1. This is the organism part, and here the column is actually what is the property I want to describe. For the sample, they have a prefix called characteristics. For the data, they have a prefix called comment, and actually this has been enforced before creating SDRF for proteomics. This is coming from the RNA and the microarray community, and we try to follow up this concept. Inside brackets, then you have the property that you want to study. In this case, for example, this is a characteristic, a disease, a characteristic, an organism, and this is the actual value for this sample. Okay, then it's clear that SDRF has some characteristics for the sample, some characteristics for the data files, and then the factor value under study. Actually, I didn't mention before, but this is extremely relevant. SDRF is an open source standard. SDRF for proteomics is an open source standard file format. You can find plenty of information and interaction with the developers of the standard here in this community, Proteomics metadata standard, and in GitHub. Then why this is experimental design? This is a really nice example of a labor-free experiment, because SDRF not only handle the metadata around the sample, the data file, and the factor value, but also the relation of the experimental design. And this is done through a couple of properties that are mandatory, required for every SDRF, that it should be written in the document. For example, and they are the label. In this case, this is really important, really different from the RNA and RNA-seq world, when you don't have multiplexing is not common. But in proteomics, you have labor-free experiment where every sample can be not labeled. Then you don't have multiple samples in the same, let's say, channel, or in the same, I would say, yeah, in the same channel. And you don't have the concept, you have the concept of replicates, but you don't, in proteomics, we have also the concept of fraction. And this is really common because also in RNA, then you have two ways of, when you do a TMT, you multiplex multiple samples in the same channel. And when you do fractionation, you split multiple samples into different rows. Then at the very end of the table, you have a row file, and then you, at the very beginning, a sample. And these three columns define how this will be combined, or how they would be related. This sample 1 with the same accession is repeated twice, and it's basically because this sample, which is the same biological replicate, it actually contains two technical replicates. This is not fractionated, but you have two technical replicates of the sample. Then if actually you can have the same relation if this is only one technical replicate, but you have two fractions. Then the relation between the sample and the data is actually defined by the technical replicate, the label, the fraction, these three things, okay, mainly. What that means is that that in the technical replicate, this means that you have repeated the same sample twice with different configurations in the instrument, and then what will be changing would be the name, the properties related with the data file. When you have two different replicates, okay, what is the concept of replicate here? Biological replicate is you have two different replicates because it's the same factor value, okay? It's the same factor value, but you have two different samples. These are two different samples for the same condition, let's say. Then they are called two different biological replicates. When you start writing the SDRF, you realize that this is the way you relate an experimental design. This is the actual experimental design. The factor value is the phenotype, and these are the sample that you have, and these are the technical replicates, and these are the final raw files. That's why, and this is really important, crucial point, this is why you have multiple, you can have repeated the sample multiple times, okay? And that's why the source name, which is the accession of the sample, is actually really important, okay? That's why the way you write an experimental design. Okay, I will not go into detail the previous slide, but I want to go into some examples. Then, for example, when you have a disease, you need to go into this EFO ontology term. Ontology we use for the sorry, we use for the sample metadata mainly EFO ontology terms. Okay, EFO is the expression experimental factor ontology, something like that. I don't really rename. I normally use the acronym, but not the full name. And it is actually an ontology that contains multiple ontologies to describe disease, organisms, for example, the cell line, and so on and so forth. And for the data description, we use EFO, but also we use MS, Unimod, and so on and so forth. Then, as I said, the actual name of the column is the characteristic and disease, and then when you go inside, the value of the column needs to be the ontology term. Let's go back to this particular example. This necrotic tissue needs to be a term in OLS. You need to go to OLS and try to find what is the term that really describes your sample for the disease. You can write the term as the name of the term, and that's the simple way to do. But we have more complex way of writing the terms, and I will explain with a couple of examples. For example, for post-translational modification, because when there is sometimes that you want to capture not only the term but also some properties of that term that is not possible to do it with the ontology term only. And this is, for example, how do you describe a PTM? And how this is related with Quantms, I will explain in detail at some point with examples, but it is important that we want to capture for every sample and every raw file what PTM, possible PTMs we are expecting in that particular data file. Because different to RNA and in proteomics, we are interested to explore what are the PTMs that are present in the sample. Of course, there will be people that say, okay, if you cannot express all the possible PTMs that are in the sample in the data file, because if you use open search, you can have multiple PTMs, not only the one that normally you look for them. That is true, but in 90% of the cases, you know really what are the main PTMs presented in the sample or in the data file, sorry, and you want to describe which are those PTMs. How do you write a key value pair? A key value pair property in the file will look like this. When you have a key, what is the key? In this case, is name of the PTM. This is the name of the PTM. In this case, is you have what is the accession in Unimod, for example, what is the target amino acid in this case, and so on and so forth. This actually is the way you describe, and you will see one big example with that. And this the way you describe the entire term and the way you describe is a key value pair with semicolon split in between each key value pair for the term. What will happen with the SDRF is if you submit an SDRF to Pride in your database, then it will go directly into another database, which is called biosample. But actually, I want to explain here where do I take the term. For example, this is organisms here, and if you go to organisms in OLS, okay, OLS, this is the parent term, and inside the parent terms, you will find Homo sapiens. And actually, this is really important because you need to have here what is the main property, and that is an ontology term in EFO, but inside you have the property of that ontology term. Okay, then you need to interact quite a lot with EFO to describe your metadata. Okay, then what we have the concept of sample templates, mainly what kind of properties are required or mandatory for each sample and for each type of experiment. For example, in human, we want for every human data set the organisms, the source name, but we also want ethnicity, age, development stage, sex, disease, organism, part, cell type, individual, and so on and so forth. If you are in a cell line experiment, you should provide the cell line code here, but not the individual, because this is not in experiments in human. As I said, this is a community-based approach, and there is a lot of people that has contributed with that. And we have a validator that people can go and then check what happened with the validator. I mean, how to validate their files created. Why this is so important? Okay, I will put one example here. Allow me to show this example. I hope this is shown in the recorded version of the tool of the presentation. This is how an SDRF look like. Okay, then here you have the samples, and here you have the properties. And why this is so important for Quantms? Quantms will take this SDRF and will split each sample into one processing node. Okay, it will take. There is some properties that are mandatory and required, like for example, the cleavage agent that was used, in this case trypsin, or for example the PTMs that was used. For example, this is how do you put oxidation, how do you write oxidation methionine, and then you put the target amino acid and the unimod. Then and also for DDA experiment, it's really important the fragment mass tolerance, the precursor mass tolerance, because this will give us how do you map, how you can identify for how you can perform the identification, which for each peptide you can map to you can identify each spectrum in the raw file. And then you have the factor value, which is in this case is the disease. This is actually all these sample and raw files are normal. And then you have the condition here, which is aquam cell carcinoma. Why actually happening with what actually happening with Quantms? Quantms will take this particular SDRF, and in the first presentation I explained a little bit about that. It will take this sample, then this raw file associated with the sample. If you have multiple samples associated, like in this case a TMT, you have multiple channels. In TMT, you need to write every channel for every sample. But if you go to the raw file, this is actually same raw file. Okay, then it will take the same raw file. Okay, and then it will trigger with the same parameters, knows what is the precursor mass, what is the fragment tolerance, and the PTMs, and it will trigger a search step, let's say with Comet, MSDF Plus, and so on. And it knows, because it knows all the experimental design, what are the channels that are there. Because of that, it performed the quantification on each channel, and at the very end, it has the experimental design to perform the differential expression analysis using MStats. Okay, and that's actually the beauty of starting from this. You describe your entire experiment. You describe all the parameters or most of the parameters that are relevant for the type of experiment. I will put another example where actually fragment tolerance are not really relevant because it's a DIA experiment. And because it has the factor value, it knows what to compare. Normal against cell carcinoma. And actually, this is a really relevant example because you have here something called norm. Norm is actually in TMT experiment, you have something like pool when you put one channel that contains all the information to perform normalization against those channels. Because this pipeline Quantms is so tight with MStats, we know that MStats, when one condition is recognized as norm, then it's used as a pool, and that's what is described and so on in that case. Then you have at the very end the quantitation, but the beauty of this is that you also have all the information about the experimental design. In this case, we don't have that much because this is a post-annotated experiment, but your own experiment, you know what is there. And this is, for example, the individual. This is P1, this is the third individual, this is also from the third individual. And then you can describe if it's a male or female, and so on and so forth. And here, the whole idea is you start by describing your experimental design, your sample metadata, the conditions, but also the properties around each raw file. And then you give this to Quantms. Quantms perform the analysis and give you back the result, which is amazing for reproducibility, for standardization, and also for making more transparent your result and your analysis. I hope this has been helpful, and you can go to the first talk to see how SDRF is coupled to Quantms. Thank you.